Please welcome Matthew Robert Huska, Max Delbruck Center for Molecular Medicine in the Helmholtz Association, exploring tumor heterogeneity in cancer.
Okay, so if you're to look inside a normal human cell, you would find 22 pairs of autosomal chromosomes and 2 sex chromosomes. But if you're to look inside a cancer cell, you'll see something very different. This is an example of a bladder cancer cell, and you'll see that whole chromosomes have been lost, others have been duplicated multiple times. We refer to this type of change as a somatic copy number aberration. Now, if instead of looking at, Now, if instead of looking at a single cell, we look at a whole tumor, you'll see that that tumor is not made up of a homogeneous mass of identical cancer cells. Instead, it's made up of a heterogeneous collection of different cell populations, each of which contains its own unique genetic background and also its own somatic copy number variants. If you consider those, along with the copy numbers you would expect to see in the normal cells, you can use these to recreate the tumor progression within that patient. Now, if you do this type of analysis on a whole cohort of patients, you can then use the structure of these trees to group your patients. So for instance, one group might have a more linear tree structure, indicating a lower tumor heterogeneity. The other group might have a more branched structure, indicating a more highly heterogeneous tumor. And if you consider the survival of the patients within these groups, you'll see that it's very different, and that we should perhaps consider different treatment options for the patients in these two groups. Now, this type of analysis involves 4 distinct steps. First, we need to identify patients and collect samples. Then we need to sequence those samples, the DNA of those samples, and then we need to actually infer the copy numbers of each segment of each chromosome using that sequencing data. These copy numbers can then be used to reconstruct evolutionary trees. Now, my work is focused on improving methods for copy number segmentation, and while other methods do exist for this type of analysis, what our method does that's unique is that it tries to determine whether or not, for every copy number change, it's the maternal or paternal copy of the chromosome that's being affected. By incorporating this information, we can generate more accurate copy numbers, which leads to more accurate trees, and hopefully allows us to better group patients for treatment and also better understand the disease. With that, I'd like to thank my collaborators at the Charité and the group of Johannes Schulte, and I'd be happy to answer questions at the break.